All right. Good morning, everyone, once again. We'll now get started with our webinar on how to visualize your research breakthroughs with RNAscope Multiplex Fluorescent Assay. To start with, we'll just quickly go over the agenda for today's webinar. We'll start with discussing the overview of the RNAscope technology, followed by the workflow for performing the assay, some guidelines on how to have successful RNAscope assays, introduction to the RNAscope Multiplex Assay on Leica Bone RX, and then this will be followed by introduction to our new product, the RNAscope HiPlex Assay, and then we'll end it with some troubleshooting guidelines on the RNAscope Multiplex Assay, how can you quantify your data, and some frequently asked questions. All right, so we'll get started with the introduction to the technology. So RNAscope assay is a specific and sensitive method to detect RNA biomarkers in cells and tissues with morphological context, or in other words, preserving the spatial resolution. It's a hybridization-based amplification, which has a unique probe design, and we'll get to that in the next couple of slides. It allows for simultaneous signal amplification and background suppression, and allows for single molecule detection. Moreover, with the RNAscope assay, you can also detect multiple mRNA markers at the same time, and the detection can be either fluorescent or chromogenic. And lastly, the assay is compatible with a variety of sample types, to name a few: FFPE, fresh frozen, fixed frozen, and cultured cells. So the RNAscope workflow begins with pretreatment, which is to unmask the target RNA and permeabilize the cell, which will be necessary for the next step, which is for the target-specific double Z probes to go in and hybridize to your target molecules, target RNA molecules. This would then be followed by an amplification cycle where you would have hybridization of amplifiers and labeled probes, and you would then detect signals from RNA molecules using a microscope. And finally, this can be quantified, or the single RNA molecules can be quantified to get the single cell gene expression. The unique part of the RNAscope technology is its probe design. So as you can see, the probe for RNAscope assay consists of double Zs. The bottom part of the double Z probe serves as the target-specific binding site, which is about 50 base pairs in length. So this region is essentially complementary to your target transcript. The bottom part is separated from a top part by a linker, and the top part is essentially what serves as the pre-amplifier binding site, which is roughly 28 nucleotides in length and serves as the base of the amplification structure. A regular RNAscope probe would be about 26 ZZ pairs in length, and therefore an ideal target region for a regular RNAscope probe would be about 1,000 base pairs in length, which would consist of 20 ZZ pairs each, that is 50 base pairs in length. Now, for most detection technologies, there are two parameters that are essential, and those are: the technology should be sensitive, wherein it can pick up signal even from low abundance molecules, and it should be specific, wherein it's not detecting off targets. So the RNAscope assay begins with binding of the double Z probes to your target sequence. This is followed by binding of pre-amplifiers on the top of the double Z probes, followed by binding of amplifiers to the pre-amplifiers, and then detection using a labeled probe, which can be an enzyme or a fluorophore. So this amplification is what provides the technology its sensitivity. The assay is also highly specific, and by that what I mean is if you had only a single Z that's binding to your target RNA, there would be no preamplifier that would be able to bind to the top of the double Zs, and therefore no signal would be generated in that scenario, therefore providing the technology specificity along with the sensitivity as we discussed earlier. The technology also allows you to look at multiple mRNAs at the same time, and this is done by a unique tail sequence at the top of the double Z probe, which is an indicative of channel designator. So as you can see on the slide, there is a tail sequence on the top of the double Z probe, which designates the tail sequence. The tail sequence for a channel 1 or a C1 probe is different from the tail sequence for C2 or a channel 2 probe, thereby giving you the specificity when you're trying to look at multiple targets at the same time and avoiding cross-contamination between different channel probes. To date, we have more than 2,600 peer-reviewed publications, which essentially shows the wide acceptance of the technology in the scientific community. If we were to break down the publications by the disease area focus, cancer and neuroscience are the disease area focus where our technology is being used the most, but that does not rule out other disease area or biological research areas such as developmental biology, detecting non-coding RNA, or looking at stem cells, to name a few. The next slide, we are going to look at a snapshot of all the different RNAscope and BaseScope assays that the company provides. I'd like to break this table into 3 halves. So we'll look at the first 2 columns, which are for chromogenic assays to start with. So for the chromogenic assays, we have 2 different options available. You can do a single-plex chromogenic assay, which can be brown for DAB HRP, or red for fast red with ALP. Both these single-plex options are available to be done manually or on our automation platforms like Bond RX and Ventana Discovery. Then you could also do duplex chromogenic staining, where you would have green and red detection via HRP and ALP. And again, the duplex assay is also available to be done manually as well as on automation platforms. We'll now focus on the column number 3, 4, and 5, or the middle part of the table, which focuses on fluorescent assays. So we have RNAscope Fluorescent Multiplex Assay, which from here on I'll be calling as version 2. We then have the version 2 of the assay, and we now have the HiPlex assay. So the version 1 of the assay, or the fluorescent multiplex assay, allows you to detect 3 targets simultaneously using Alexa 488, 80550, and 80647. And this, at this point in time, is only available to be done manually. Whereas the version 2 of the assay has TSA-based amplification and background suppression, allows you to look at 4 mRNA targets simultaneously, and is available on both manual as well as Leica Bond RX platform. And then we'll go on to our newest product on the market, which is the HiPlex assay, which allows you to detect 12 targets simultaneously on a single tissue section. And at this point in time, the HiPlex assay is available only to be done manually. And then the last 2 columns are looking at the BaseScope assay, which is designed to detect shorter sequences or sequences that are 300 nucleotides or lower in length. The BaseScope at this point in time is available as chromogenic singleplex or chromogenic duplex, and only the singleplex assay at this point in time is available on automation platforms such as Leica Bond RX and Ventana Discovery Ultra. As you know, I just would like to reiterate the fact that the focus of today's webinar is going to be on the multiplex fluorescent assays, so we'll focus on those for today's webinar. We'll now look at the assay workflow. So the assay essentially can be done in a single day, or it can be split up into 2 days to make it a 2-day assay. As you can see on the left, for a 1-day assay, you would follow it with sample preparation, followed by pretreatment. This would then be followed by probe hybridization and detection of the signal. You can also split up the assay into 2 different days using 2 different options shown here. You could stop on day 1 after sample pretreatment and continue with probe hybridization on day 2, or you could use option 2 for a 2-day assay wherein you would stop after probe hybridization and proceed with the detection of the signal on day 2. We'll now look at the workflow in a little more details for the different multiplex fluorescent assays. We'll start with the fluorescent multiplex assay, or the version 1 of the assay. The workflow shown here is for fresh frozen tissues. So as I mentioned previously, all RNAscope assays would start with pretreatment, which in this case is performing a post-fix and a protease step, which is essentially required to chew up your extracellular matrix, which is important for your multiplex probes to go in and hybridize to target RNA. The probe hybridization will be followed by a series of amplification steps, AMP1, 2, and 3, which would then be followed by using a AMP4 alt combination, which would be for a labeled probe. This would then be followed by DAPE to counterstain and mounting with prolonged gold and cover slipping the slice. Please note that out of the 3 AMP4 alt combination, you should choose only one of the 3. And in the next slide, I'll go in a little details about what those 3 combinations are. So the 3 different alt combinations for AMP4 with the version 1 of the kit essentially gives you 3 different color module configurations. For the purpose of simplifying the table, the best way to look at it is if you look at Alexa 488 or the green fluorophore with Alt A, it's in channel 1, in Alt B, it's in channel 2, and it's in channel 3 for Alt C. One other important point to note here is with the version 1 of the assay, we at this point in time do not have a far red fluorophore or 80647 that can be associated with a channel 1 probe. We'll now look at the workflow for the version 2 of the multiplex fluorescent assay. The workflow shown here is for FFPE or formalin fixed paraffin embedded tissues. Again, the pre-treatment starts in this case because it's FFPE, starts with baking and deparaffinization to remove the wax, followed by H2O2 step, which is required to degrade the endogenous peroxidase, since this is an HRP-based reaction, followed by target retrieval to reverse the fixative crosslinks, followed by the protease step to degrade the extracellular matrix. This would then be followed by hybridization for the multiplex probe, and then a series of amplification steps, AMP1, 2, and 3, and this would then be followed by sequential development of each channel probe. So the first fluorophore, you will start with HRPC1, followed by incubation with opal dyes or TSA plus fluorophores. Please note the opal dyes or the TSA plus fluorophores have to be purchased separately from Akoya Biosciences. And after that, you will do the HRP blocker step to stop the reaction for that particular fluorophore, and you essentially repeat the steps for the second channel probe and the third channel probe, followed by DAPE for counterstaining and mounting with prolonged gold and then cover slipping. A very important point to note is irrespective of the channel probes or the number of targets that you're probing for with the version 2 of the assay, AMP1, 2, and 3 steps are mandatory because these 3 steps would sequentially build the amplification tree for all channel probes. Or in other words, if you're only doing a channel 1 probe with the version 2 of the assay, it is still mandatory to perform AMP1, 2, and 3 steps. Now, one of the major questions that would arise when you have multiple options is which of these multiplex fluorescent assays is right for you. So the table here demonstrates compare and contrast between different multiplex assays that we have. As I discussed previously, we have the version 1 of the assay, version 2 of the assay, both of these that could be done manually. Then we have the version 2 of the assay on the Leica platform, so that's LS multiplex fluorescent assay in column 3. And then finally, we now have the HiPlex assay in column number 4. So some key points of differences between these assays is the version 1 of the assay does not have the TSA-based amplification like the version 2 of the assay or LS multiplex assays have. And this is important if you're working with a low expressing target or tissues that have high autofluorescence. In that case, it would be better to use the version 2 of the assay. And the dyes used, of course, are different for this assay. As I mentioned previously, the version 1 of the assay uses the Alexa and the Auto-fluorophores, which are directly conjugated. The multiplex fluorescent, either manually done or on Leica platform, uses opal dyes with TSA plus fluorophores, and then the HiPlex assay uses Alexa fluorophore 488, 550, 647, and 750. One another key point of difference between the assay is the number of targets that you can probe for simultaneously. Version 1 of the assay allows you to do only 3, whereas multiplex fluorescent V2 or LS multiplex fluorescent on Leica allows you to do 4 targets. And then the HiPlex assay allows you to do up to 12 targets simultaneously. With regards to the compatibility of the assay with immunostaining or IF, we do not recommend using the version 1 of the assay. However, the multiplex fluorescent V2, LS multiplex fluorescent, and HiPlex, all these 3 assays are compatible for dual-ish IF. Please bear in mind, though, that the protease step during the assay may negatively impact the protein expression. And then the final point of difference would be the sample types that are compatible. With the V1 assay, FFPE sample type is not compatible, so you can do either fresh frozen, fixed frozen, or cultured cells. With the version 2 of the assay, all the sample types are compatible. FFPE, fixed frozen, cultured cells, fresh frozen, to name a few. Similar is the case with LS multiplex. And then with HiPlex, at this point in time, you can only use it to do fixed frozen and fresh frozen sample types. All right, so we'll now go into some tips on how to get started with the RNAscope multiplex assay. Shown on this slide are pictorial representation of the reagent kits from ACD. So in order to do the assay, you will need pretreatment reagents, you will need the detection kit that has the amp, and then you will need the wash buffer to do the washing steps. Please note, the wash buffer is provided at 50x and would have precipitation. And in order to dissolve the precipitate, we recommend that you warm the wash buffer at 40 degrees for 20 minutes, which will dissolve the precipitate. And if you're trying to do a 4-plex assay using the version 2, then please note RNAscope 4-plex ancillary kit is required. Here are some of the recommended accessories that we recommend to be used with the RNAscope assay. So you would need the easy batch slide processing system, the hybridization oven, which is essential to the success of assay because it helps maintain optimal temperature and humidity. You will need the tissue tech washing tray, and then you will need either a hot plate or a steamer to perform the target retrieval or the antigen retrieval step. It is essential and highly recommended that you review the checklist before getting started to make sure that you have all the required materials and reagents. Just to point out a couple of important factors on the checklist, please ensure that you use SuperFrost Plus Light. Ensure that you use the high BZ oven because it helps maintain optimal humidity and temperature. And then always ensure to run positive and negative control probes to determine the RNA quality in background. Ensure that you follow the workflow guidelines. To emphasize on a couple of guidelines, please make sure that you perform all the M steps in the right order and do not skip them. Also make sure that you do not let the slides dry out unless it is specifically mentioned in the protocol. And it is preferred that you use fresh reagents such as ethanol, xylene, or formalin for fixation if possible. Here are some of the key guidelines for performing the RNAscope assays. To start with, please ensure that you follow the protocols as exactly described in the user manual. Please make sure that you review the sample pretreatment conditions for your sample type. The pretreatment differs based on the sample type, and we'll get to that in a bit. Always use controlled probes to confirm the RNA quality for your samples, and then make sure that the negative control gives you no background or non-specific signal. And then finally, ensure that you review the checklist to confirm that you're using all required materials. I'll now go through sample preparation guidelines on a couple of different sample types. We'll start with FFPE or formalin fixed paraffin embedded samples. FFPE samples, we recommend that they're fixed in 10% neutral buffer formalin for 16 to 32 hours at room temp, followed by standard processing and embedding. These sections should be cut at 5 plus and minus 1 micron in thickness, and they should be mounted on SuperFrost Plus Light. For storage, we would recommend that you store the sections with desiccants either at 4C or room temperature up to 3 months. If you are going to store them for longer, we suggest that you store them at 4 degrees Celsius with desiccants to preserve RNA quality. Please note, the FFPE samples at this point in time are only compatible with the multiplex fluorescent V2 or the LS multiplex fluorescent assay. We'll now look at the fixed frozen sample preparation. So for fixed frozen sample types, we recommend fixing them in 4% PFA at 4 degrees for 24 hours, followed by embedding them into OCT. And then you may store the block at -80 for up to 3 months in an airtight container. For sectioning, for fixed frozen samples, we recommend a section thickness of 7 to 15 microns. Again, please note that you use SuperFrost Plus Light. Air dry the slides at -20 for 1 to 2 hours before you move them to -80 degrees Celsius, and try to see if you can use these sections for the assay within 3 months of storage. In the taking the time into consideration, I'm not showing the sample preparation for fresh frozen and cultured cells, but please note the assay is compatible with those two sample types as well. Now, as I mentioned previously, the sample pretreatment recommendations would vary depending on the sample type, and shown are those differences for different sample types on this table. So for FFPE samples, since they are drop fixed in formalin and room temperature, they would require antigen retrieval or target retrieval to reverse the formaldehyde cross-links, and Protease Plus, which is our weakest enzyme. Fixed frozen samples, since they are fixed at 4C, would require shorter target retrieval time, so that's only 5 minutes, and a stronger protease, which is Protease 3. Fresh frozen samples, since they did not get any fixation or they were not drop fixed, did not require target retrieval, but they would get the strongest protease, which is Protease 4. And finally, for cultured cells, again, since they are not drop fixed like FFPE or fixed frozen samples, no target retrieval is required, but you will use Protease 3 with appropriate dilution because cells, unlike tissues, do not have a lot of extracellular matrix that needs digestion. Okay? Now, we highly recommend that before you begin your assay, that you qualify your samples using controls. An ideal workflow would be to start with control slides. Shown in this scenario is HeLa control slides. You can also get 3D3 control slides if you're working with mouse tissues from us. So the first point would be to perform a technique check where you would do 3-plex negative and positive control probe staining on control slides, ensure that there's no background for the negative control probe and that there is robust staining for the positive control probe. I would like to emphasize on the fact that the negative control probe staining is very critical when performing fluorescent assays, because if you were to use a higher exposure setting, you may pick up the background or non-specific staining in the negative control probe. So when imaging, ensure that the negative control probe is clean, and then use those same image acquisition parameters to image the positive control probe staining or target probe staining. Once you perform the technique check using the control slides, the next would be to do a sample RNA quality check. So you would run those negative and positive control probes on your target tissue. Once the target tissue passes QC, where the RNA quality is good, then you may go on to run your target probes. If this fails, then we recommend that you verify the technique using control slides, check RNA quality with new samples, and perform optimization if needed. The next couple of slides, we will look at some of the staining examples for the fluorescent assays. Shown on this slide is a 3-plex staining using the fluorescent multiplex V1 assay. This image is taken from a publication where we're looking at staining for FABP5, MIF, and FCRLS in fresh frozen mouse cerebellum from a P5 pup. Again, remember, the version 1 of the assay is recommended for fresh frozen or fixed frozen sample types. Please note that all channel 1 or C1 probes are provided as 1X or ready-to-use format, whereas C2 and C3 probes are 50S. Therefore, the C2 and C3 probes should be diluted 50 times using a C1 probe before use. If you plan to use only C2 and C3 or only either one of those, then you may use a probe diluent to dilute those channel 2 and channel 3 probes 50 times. Finally, please note prolonged gold is the recommended mounting media for the fluorescent assays. Shown on this slide is a 4-plex staining using the multiplex fluorescent V2 assay in mouse brain coronal sections. In this scenario, we are looking at staining for 4 different markers. As you can see on the right, for VGAT, GAPDH, VGLUT1, and VGLUT2. Again, prolonged gold is the recommended mounting media. We recommend that you start with TSA plus fluorophore or opal dye concentration at 1 to 1500. And please note that this fluorophores need to be purchased separately from Accoya Biosciences. We'll now switch gears and go from the manual platform to the automated platform. So we'll now look at how you can perform your RNAscope assay on Leica RX for automated-ish detection. Shown here is an overview of the Leica workflow. Please contact ACD sales or Leica sales to schedule RNAscope training and for software updates. And please note we have field application scientists available for troubleshooting on the Leica RX platforms. Shown here is the workflow for the LS multiplex fluorescent assay. This can be essentially divided into three major parts. The first part would be essentially setting up the protocol, where you register the reagents, create the protocol, create a staining protocol. Please remember, a staining protocol needs to be created once for each probe. Register the mark probe, dilute the fluorophores. Once the protocol setup is done, you will then go on to set up for the assay. So you would set up a study for the multiplex assay, run the assay, which would include pretreatment, probe hybridization, amplification, detection, and counterstate, and then finally mount the slides with prolonged cover slip. And then the final step, of course, would be to image or scan the slides to look at the results. The LS multiplex assay, the runtime is about 14 hours. Please note that the kits provided are enough to stain 60 slides, whereas the probes are sufficient to stain only 30 slides. So please keep that difference in mind. And please note that a bond research detection system from Leica would be required for performing the LS multiplex fluorescent assay. Again, signal detection is performed using opal dyes or TSA plus fluorophores, which need to be purchased separately from Akoya Biosciences. And then if you're doing a 4-plex detection, then a 4-plex ancillary pack will be required. Shown on this slide is a pictorial representation of the interface on Leica Bond RS software 6.0. And essentially, there are different regions on the interface that are marked. You have options for setting up the slides, setting up the protocols and reagents. You can toggle between the two instruments shown on the left. And then you would have slide trays represented here. And on the bottom right, you have indication on what is the volume status for your bulks. And finally, on the top of that, if you have errors, you will have instrument notifications. Shown on this slide is the slide setup for 5.2 and 6.0 software on Leica Bond RX. As you can see, the top part is where you would have description about the slides. And then the bottom part is essentially choosing the protocols. So you would choose the staining protocol, preparation, for example, bake and dewax if you're working with FFPE samples, and then you will choose the standard heat-induced epitope retrieval and enzyme treatment. And on the right is shown the 6.0 setup. Please note when you're using the 6.0 setup, the probe application and probe removal should always be set as default. On the next slide, we will look at an example of a successful staining using the LS multiplex fluorescent assay. We're looking at 4-plex staining, and we're looking at immune cell infiltration in non-small cell lung cancer tumors here, which are FFPEs. Shown here is 4-plex staining for CXCL9, CCL5, interferon gamma, and CD8. On the top right image, the blue arrow marks a cell that is positive for all the 4 markers shown here. Okay, we'll now again switch gears and look at our newest product on the market, which is the RNAscope HiPlex assay. RNAscope HiPlex assay essentially is our newest product, as I mentioned, and allows you to detect 12 RNA targets simultaneously. The product is available in 2 different configurations. You have the HiPlex 8 reagent kit, which allows you to do the first 8 targets, which can be upgraded with an ancillary kit to do a 12-plex. Please note that this assay is enough for performing staining on 10 slides, and that all the probes are provided as 50x concentrate, which should be diluted using a HiPlex probe diluent, which is provided with the kit. And finally, you will have the HiPlex registration software that would be provided with the kit as well. An important point to note is that the HiPlex probes are not compatible with the existing reagents for the multiplex fluorescent assay or vice versa. Some of the key highlights for the HiPlex product: at this point in time, it's only going to allow you to do manual detection. It allows you to do 12 mRNA targets on the same tissue section. And it's a simplified workflow with a single probe hybridization. Please note that the detection is iterative, wherein you will do 4 targets at a time in 3 rounds to detect a total of 12. The assay utilizes cleavable fluorophores, which are Alexa fluorophore 488, 80550, 80647, and Alexa fluorophore 750. The assay, like the other RNAscope assay, conserves morphology and preserves RNA quality with chemical-based rapid cleaving. No target probe stripping is involved, and at this point in time, it is recommended only for fresh frozen and fixed frozen sample types. Please note that the unique part of the RNAscope assay, which is the double Z probe design, still remains the same. And we'll next look at the signal amplification for the HiPlex assay. So the HiPlex assay starts with hybridization of all the 12 probes, which would be T1 through T12. This would then be followed by amplification tree built on all the 12 different fluorophores, all the different 12 channel probes. And just like the regular RNAscope assay, the assay is very sensitive as well as specific, wherein if you only had a single Z bind, you will not generate a signal. So the assay starts, as I mentioned, is going to be in 3 rounds of detecting 4 targets at a time. So you will start with detection of T1 through T4 to start with. This is going to be followed by chemical cleavage to get rid of the fluorophores only. Please note only the fluorophores are cleaved here via chemical process, and the probe hybridization of the tree is not going to be destabilized. You would then detect the next round, which would be T5 through T8. Again, this would be followed by chemical cleavage, and then you will perform detection for the final round for T9 through T12. Shown on this slide is an overall view of the RNAscope assay workflow. So day 1 or round 1 is where you will spend most of your time, because that's where the target probe hybridization and signal amplification happens for all the 12 probes simultaneously. You would then do T1 through T4 detection. So that would be imaging 1. In round 2, you will do detection for T5 through T8 after cleaving the T1 through T4 fluorophores, and then you would go on to imaging for round 2. And then finally, if you're doing a 12-plex assay, which would require the HiPlex ancillary kit, you would cleave the T5 through T8 using a chemical cleavage, followed by T9 through T12 fluorophore detection and image for round 3. And finally, you will use our image registration software to basically stitch the images from round 1, 2, and 3 together to create a merged image. I'll now show some of the applications of the HiPlex assay. So shown on this slide is an application of HiPlex assay where you're doing RNA-seq validation for neuroscience. And it shows beautiful distinction between staining for different targets in the ventral striatum and basal forebrain from mouse. So again, the staining looks great because you have multiple targets here, but also note that it's preserving the morphological context. Now shown on this slide is the HiPlex 8 staining for all DRD1 alpha subpopulations in the brain. And on this slide, we're looking at the DRD2 populations in the brain. And finally, shown on this slide is all the different subpopulations detected by HiPlex 12 probes. Please note the figure shown here is a representative of a 40x field of view and may not show all the populations. So again, this is just to show the power of the HiPlex assay to show that it is capable of labeling 12 mRNA targets at the same time. We will now look at some of the troubleshooting tips for the RNAscope Multiplex assay. The key question to ask is, why is it necessary to optimize the assay? So oftentimes you would have samples that would be underfixed or overfixed, or you would be working with sample types such as xenograft cultured cells, which would require different pretreatment, or you would be working with different tissue types such as liver, muscle, or retina, which again would require different sample preparation or sample pretreatment especially. So how would you perform the pretreatment optimization? So depending on the issue at hand, so if you have underfixed tissues or overfixed tissues, you may adjust the boiling time or the target retrieval time or the protease step to basically counter for under or over digestion. One of the most common issues that you would see with the fluorescent assays is the issue with autofluorescence. And shown here are a few examples of autofluorescence from different tissues such as kidneys, intestine, brain, and colon, to name a few. Please note, if you see autofluorescence, then you may adjust the TSA fluorophore or the opal diode concentration. We recommend that you start with 1 to 1500, but if required, you may change the concentration. If you're seeing autofluorescence, we recommend that you assign the highest expressing target with the green channel or the green fluorophore, which would be opal 520 or Alexa 488 for the version 1 of the assay. And then finally, you may use multispectral imaging software to subtract background autofluorescence. One of the other issues that need troubleshooting would be sample digestion. Please note, over-digested samples will tend to give higher autofluorescence. So as shown in the picture here, on the top left, you see an over-digested tissue where you're seeing high autofluorescence, but when you use the optimal conditions, you're able to see better signal-to-noise ratio, or in other words, you're able to see distinct signal from the background. So the tips would be to use the recommended protease as per your sample type, and make sure that you don't over-digest your sample, which would cause autofluorescence and may mask your staining. Now, this table shows some of the common fluorescent issues. So in scenarios where you would see no signal or weak signal, there are 3 major potential sources or root causes for that issue. It could be the microscope, the assay itself, or the sample type. So please make sure that you have the correct filters on the microscope for imaging the fluorophores. Please make sure that you're not overexposing your tissues or using the wrong exposure. So make sure that you verify the signal with the naked eye first before using the auto exposure. And then please make sure that you're using the appropriate image enhancing software such as Nuance. The second potential source would be the assay. So if you were to skip the M step, so as I mentioned previously, please note completing all the 3 M steps is mandatory regardless of the number of targets that you're probing for. So make sure you do all the 3 M steps. And then finally, it could be the sample where you can have RNA quality compromise. If that's the case, please get fresh samples with good RNA quality. You could have issues with sample preparation. So in those cases, please make sure you follow our pretreatment and sample preparation guidelines. Please perform the 3-plex control and 3-plex negative control probe to assess the RNA quality of the samples, and then make sure there's no background with the negative control probe staining. And as I mentioned previously, always check the signal with naked eye under the objective lens first. Another parameter that might require troubleshooting is sample preparation and detachment. So there are 2 probable causes where you would see sample detachment. One would be if you were to use wrong slides. So please make sure that you use Superfrost Plus slides. These slides are charged and help ensure that the tissue does not detach during the assay. The other reason could be suboptimal tissue preparation. So if the tissues were underfit, then there's a chance of detachment. So please make sure that the samples are prepared according to our recommendations. If required, you can bake the slides for longer and follow the recommended tips for solutions. I'll go through in the next couple of slides tips on how you can avoid sample detachment depending on the sample type. So for FFPE sample type, if you're seeing sample detachment, then you may bake your slides for 30 minutes at 60 degrees Celsius after deparaffinization and after target retrieval as well. Also, please note that we recommend that you bake or dry the slides in an active air circulating oven and not the high BZ oven preferably. If you're seeing sample detachment with fixed frozen sample types, then here are the 4 recommended troubleshooting tips. The first tip is ensure that you are air drying the slides for -20 for 2 hours. You may post-fix the slides in 4% PFA for 15 minutes to 2 hours at room temperature. Bake the slides after target retrieval for 30 minutes at 60 degrees Celsius. And in scenarios where step 1, 2, and 3 or tips 1, 2, and 3 do not work, you may omit the target retrieval and extend the protease time to 45 minutes. But please note that is only if the modifications 1, 2, and 3 fail. The other troubleshooting parameter would be for probes. So in certain scenarios, for example, if you're working with knockouts, please ensure that the probe is targeting the region that is knocked out. And if you're trying to detect null genes or you're trying to get probes for genes that are not already available in our catalog, then you can have custom probes designed using the link shown on the bottom right to have made-to-order custom probes designed. And then finally, if you're working with unique sample type, it might require some troubleshooting. So for example, if you're working with sample types such as whole mount, free-floating, or bone samples, it might require additional troubleshooting guidance. And we request you to contact technical support using the email address shown at the bottom of the slide for recommendations. So these were some of the troubleshooting tips for the multiplex fluorescent assay. We'll now look at the dual-ish ISC or IF application for the RNAscope followed with immunostaining. Please note the workflow is similar. So you will proceed with sample fixation, pretreatment, probe hybridization, and signal detection. So in other words, you will start with the RNAscope assay first. So some of the general recommendations for the dual-ish IHC or IF are: make sure you only combine an optimized RNAscope protocol with an optimized IF protocol. Please note that all dual-ish IHC or IF protocols would require optimization. Ensure that you proceed with the RNAscope first, followed by immunostaining, and you may optimize the IHC or IF separately using RNAscope pretreatment reagents. And finally, there are 2 points to remember: that dual-ish IHC or IF works better for highly expressed proteins. This is because of the fact that the protease treatment may negatively impact the protein expression. So if you are trying to combine it with immunostaining, we would recommend that you combine it using a protein target that's relatively high in abundance, and make sure that you use validated antibodies or validated IHC or IF protocol. The next slide, I will show an example of a dual-ish IF staining. So on this slide, we are looking at dual-ish IF for 3-plex staining using RNAscope for VGAT, VGLUT1, and VGLUT2 mRNA. This is combined with the immunofluorescence staining for neoantigen protein in green. And shown on the right are the zoom-in version for the staining for individual targets. Here's another example of dual-ish IF staining for tumor cell infiltration in cervical cancer tumor microenvironment. We're looking at PD-L1, PD-1, and interferon gamma staining with RNAscope, and CD3 staining using an antibody. The blue arrow indicates the cell that is positive for PD-L1, and the yellow arrow indicates cells that are positive for interferon gamma and CD3 plus T cells. We will now look at some of the guidelines for performing RNAscope quantification. So some of the key questions with regards to the signal are: what does a dot mean? What is the significance of a dot size? And what is the difference between a dot and a cluster? And I will try to answer those questions or define those questions in the next slide. So shown on this slide is how you can distinguish between dots and clusters. Please note mRNA molecules are detected as puncted dots, shown by yellow arrows, beg your pardon, on the slide. Please note each dot represents a single copy of mRNA. If you have signals coming from multiple mRNA molecules that are very adjacent to each other, then you may have overlapping signals, which could result in clusters, shown by the white arrows here. And another key point to remember is the dot size and the intensity can differ depending on the number of ZZ probes that are bound to the target RNA molecule. So if you look at the 2 yellow arrows pointing to 2 single dots, you can clearly see the difference in the size and the intensity of the dot. But the important point to remember is that it's the number of dots that matters and not the size or the intensity, because each dot represents a single copy of mRNA. On the next slide, we will look at some guidelines for quantifying. So shown on this slide is how we can do semi-quantitative scoring for the RNAscope assay. So you can have scoring from a 0, which goes all the way to 4. So 0 would mean no staining or less than 1 dot per 10 cells. This would be then going all the way up to a score of 4, which would be more than 15 dots per cell and more than 10% dots in clusters. Please note, you can also quantify the RNAscope assay using ImageJ or cell profiler, and we have technical notes for performing those data analysis using ImageJ and cell profiler. With that, I come to the end of today's webinar. Shown on this slide is the contact details for ACD technical support. You can contact us via email at support.acd@bio-technique.com, or you can contact us via the phone number mentioned on the slide and then choose option 3 for technical support. Please note, we are available to talk to you or help you via the phone from 8 a.m. to 6 p.m. Pacific Standard Time. And finally, you can go to the link at the bottom for support-related resources. Again, my colleagues are still on the line, and they would be more than happy to answer any questions that you may have. And I would like to re-emphasize on the fact that you should direct all your questions to all panelists in the Q&A window. We'll now go through some of the frequently asked questions. Hello, everyone. I've just been informed about the discrepancy in the timing for the webinar start time, which was listed, unfortunately, as 9:30 p.m. Pacific Standard Time. I truly apologize for the inconvenience for our attendees. However, I would like to let you all know that the webinar is going to be posted on our website. So the recorded webinar will be available for viewing, and we will send out a link to all of you for the recorded webinar. But once again, apologies for the confusion with the start time. Towards the end, I would just like to let you all know about a upcoming webinar from Protein Simple on a single platform technology for protein and nucleic acid biomarker analysis in non-small cell lung cancer. This is going to be presented by Dr. Latham, who is the Senior VP of R&D at Assays, and this is scheduled for 9 a.m. Pacific time on February 20th. Shown at the bottom of the slide is the registration link, and I would urge all of you who would be interested in learning more about this webinar to register. Finally, here are the contact details again for technical support. Once again, I apologize for the confusion with the webinar start time. However, please note my colleagues are still on the line, and they would be more than happy to answer your questions. Please direct all your questions to all panelists in the Q&A window, and they would be more than happy to help. Once again, I would like to thank all of you for attending today's webinar.